In this video, we will discuss about role of CCL5 in tumor development through innervating neuron. So before going directly into the topic, there are some more results that are presented here that is related to the ATF4 and SLIT2. So in the panel K, it is about the SLIT2. So basically, they are checking the effect on this tumor-associated macrophages because of the absence of SLIT2. And what they are able to see in the control, they can see that there is a tumor suppression when they use the chemogenetic inhibitions. And however, uh, in the case of control plus the injection of recombinant SLIT2, there is also a tumor suppression. But what they are able to see that without chemogenetic inhibitions, there is a significant increase in the tumor volume. And but when they use this slit to knock out, the interesting thing that they are able to see, and this is what they are expecting, that there is no difference with and without chemogenetic inhibitions on this tumor associated macrophages. However, when they reintroduced regionally reintroduced this recombinant SLIT2 in the SLIT2 knockout, they are again able to see a significant difference between the tumor-associated macrophages. That is, after chemogenetic inhibitions, this tumor-associated macrophages goes down compared to the control case. And similar results they are able to see for the ATF4 knockout. That is, in the case of ATF4 knockout, there is no significant difference between the TAM with and without chemogenetic inhibitions. However, when they uh, reintroduce recombinant SLIT2, they are able to see a significant difference, significant suppression of the tumor volume after chemogenetic inhibitions. So this is about the importance of, or this is about the role of SLIT2 and ATF4. That is, both of them are necessary for this tumor suppressions in the presence of chemogenetic inhibitions. Afterwards, they wanted to find out, or they investigated a they investigated this ligand pair that has the highest interactions between the resident immune cells in the tumor-derived lymph node and the tumor-associated macrophages. And from the interaction pairs, they are able to find out that it's the CCL5 that has the highest interactions. So they investigated based on this CCL5. So first they checked the CCL5 level in the stereosync analysis at different conditions, and they are able to see that in the case of chemogenetic inhibitions, there is the highest expression of this CCL5 compared to the control and compared to the other conditions. And afterwards, they also check this cytokine analysis. In this case, also they are able to find out that there is a high expression of the CCL5 in the presence of chemogenetic inhibitions, and also in the uh, in the conditions where CALSA gene is knocked down, it is responsible for CGRP protein. So it will also prevent these interactions of downstream signaling of this activation of innervating neuron. Both of them cause the high expression of CCL5. And once getting these positive results from two different studies, they further checked the CCL5 level in different conditions. Because in the wild type conditions, they are able to see CCL5 levels to a baseline. However, when they use the tumor progression model, that is optogenetic based calcium concentration increase in the intercellular part. In this case, they are able to see a decrease in the CCL amount. However, when this system is combined with the tumor suppression model, it is using this HM4 DGA, there is a rescue of the CCL5 levels are shown, and this indicates the CCL5 has a direct role towards tumor suppressions. Thereafter, they checked in the CCL5 knockout conditions. They do not see any tumor change with and without chemogenetic inhibitions. This is what they expected, and even the TEM levels does not change in these conditions. Afterwards, they checked it. They checked it in the conditioned media. In the conditioned media from this tumor-derived lymph node. In this case, also they are able to see the same results. That is, once the CCL5 has been knocked down, with and without chemogenetic inhibitions, they are not able to see any tumor suppressions. And however, there is an increase in the tumor volume compared to the case where they don't use chemogenetic inhibitions and when the CCL5 is knocked down. That can be seen here. And similar results are observed for similar trends are observed for the tumor associated macrophages. This goes up compared to the wild type case where CCL5 is present and the CCL5 is absent. In the absence of CCL5, the tumor volume goes up. And thereafter injected with a different model where they increase the amount of CCL5 using murine CCL5, and in this case also when the CCL5 amount are increased compared to the control, they are able to see a decrease in the tumor volume. However, when they checked it with and without chemogenetic inhibitions within this murine CCL5 conditions, they do not see any significant changes. And similar trends are observed for the tumor associated macrophages also. So this in all together indicates that the CCL5 also CCL5 interactions also have a role towards this tumor development through this through the activation of the innervating neuron. And basically higher amount of CCL5 will help in the tumor suppressions. So this is what they finally conclude, not only this figure but also in the whole paper, that CCL5 also has a role towards this tumor development through the activation of innervating neuron, along with the other things that we have discussed previously about the results mentioned in this paper. That's all for this video. If you need to understand more about this paper and these experiments in detail and wants to refer to the supplemental figures, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.